and did his PhD there. After that, he jumped over the pond to London and did a postdoc at Birkbeck College. After that, he learned his craft in fragment-based drug discovery at Aztecs. And in 2007, Rob joined the Institute for Cancer Research as a joint position between the Center for Cancer Drug Discovery and the Structural Biology Group. And since 2010, you lead the hit discovery and structure design group. And I think Rob's team has over the years really contributed many seminal papers in the area of structure-based drug discovery. And I'm looking forward to your talk, which is about challenges and opportunities in small molecule cancer drug discovery. And with that, Rob, the floor is yours.
Okay. Thank you very much for that kind introduction, and thank you very much to the organizers for inviting me to give this this talk here. So challenges and opportunities in cancer drug discovery was quite a big topic, so I decided to focus it a bit, and I'll tell you a bit about the cancer drug discovery through the lens of our organization. And our organization does small molecules, so it will be focused on small molecule drug discovery. So as an outline, if I can actually change the slides. Yeah, okay. I will introduce a little bit the ICR to give you a bit of context, the Center for Cancer Drug Discovery within the ICR. I will also go into how we adapted our organization to the challenges in cancer drug discovery that we're facing. I will sort of show the power of structural biology into structure-based drug design samples from our institute. Talk a little bit about how in my group we deploy cryo-EM in cancer drug discovery, and in the light of the panel discussion, I'll have a one-slider on Alphafold and AI in structural biology for structure-based drug design. So the Institute of Cancer Research is a world-leading cancer research institute. We are a charity, and we're about 1,100 staff and sort of grouped in 90 research group. We're a member institution of the University of London, and that means we can actually train PhD students, and we've got about 160 PhD students to actually train them to be the next leaders in cancer cancer research. Our research takes place at 2 laboratory sites, one in central London in Chelsea and one south of London in Sutton in Surrey. The mission of the ICR is to make discoveries that defeat cancer, and we do basic translational and clinical research that is actually grouped in 8 research divisions, and Tobias already said that. My group is actually in 2 divisions, cancer therapeutics and structural biology. What is very important to us is the unique partnership that we have with the Royal Marsden NHS Foundation Trust. The Royal Marsden is the oldest cancer hospital in the world, and they're co-located both on the Chelsea site and the Sutton site with the ICR. So the Center for Cancer Drug Discovery at the ICR is the drug discovery arm of the ICR. It is cancer drug discovery embedded in an academic institute, but we very much operate like a biotech. Its mission is to discover novel small molecule therapeutics for the treatment of cancer and progress them to hypothesis testing phase 1 clinical trials. Now, we capitalize on the science of the ICR, and we generate small molecule therapeutics with a range of modalities for high-risk targets. We have a bespoke building now, which you can see in this photo, very nice, on the Sutton campus. We're about 150 staff, and we have all the skill sets to do state-of-the-art modern drug discovery. Again, as I said earlier, the partnership with the Royal Marsden Hospital is very important to us because it gives us feedback on our drug discovery from the clinic. We work a lot with industry, and we partner at the best time and opportunity for a project with patient benefit as the main driver. So challenges and opportunities for the CCDD. Well, we are sort of a mid-sized biotech. We run a portfolio, drug discovery portfolio that is sort of similar to a mid-sized biotech. So for us, it's very important to select the best biological target. We also want to identify a high-quality hit matter tailored to the modality of the target modulation. And of course, we want to select the optimal clinical candidate and carry out, design, and execute the right clinical trial. So to help us with selecting the best biological targets, we recently have established the Center for Target Validation. And this is bespoke, ring-fenced resource that capitalizes on ICR basic science and helps to connect it to the drug discovery. The focus is on robust target validation, and it informs on the optimal method of modulation, for example, inhibitors versus degraders. Also, it gives us a first assessment of ligandability and druggability of a target. Now, the second center that we established was the Center for Protein Degradation, and that helps us to expand on the modalities by which we actually can target a cancer target. So its aim is to generate degrader tools to help unravel cancer biology, to discover new therapeutic approaches that exploit targeted protein degradation for cancer patients, and to develop new protein degradation approaches. Well, sort of in a kind of cartoonish picture, you can see that these two centers really sit between the CCDD and the ICR cancer biology, and it helps us to connect everything fluently. And then, of course, on the other side, we have the Royal Marsden Hospital, and we connect with that. So how do we identify high quality hits? So we have a variety of compound libraries for a diverse range of targets. We have fragments, small libraries. We have a raft of focused library sets ranging from about 500 compounds to about 6,000 compounds, like the molecular glue libraries that we now have. We have a 200K HTS library that is sort of a larger library and of somewhat larger compounds. And very recently, we also implemented DEL screening in my group, actually at ICR. That is, of course, a vast and very diverse library of bigger compounds. We screen these libraries with a variety of biochemical, biophysical, and cell-based assays in a high throughput formats using state-of-the-art dispensing plate reader technologies. This allows us to target a range of different target classes and targets, of which you see some of them that we targeted in the past here. So obviously, everything is underpinned by structural biology, X-ray crystallography, including XChem crystallographic screening at the XChem facility at Diamond, cryo-electron microscopy, and all of that is again underpinned by recombinant protein production, and we can do E. coli, insect cell, and mammalian expression. So how does this work in practice? I'd like to highlight that by our project to inhibit monopolar spindle 1 kinase, MPS1, or by its other name, TTK, which is a dual specificity kinase critical for the spindle assembly checkpoint. It controls chromosomal alignment and kinetochore microtubule attachment, and high MPS1 levels correlate with aggressiveness of cancer and poor patient survival in glioblastoma, PDAC, and triple-negative breast cancer. So inhibition of MPS1 induces premature mitotic exit, gross aneuploidy, and cell death in human cancer cell lines, and that includes triple-negative breast cancer. You can see its structure here on the right-hand side, typical kinase structure with a C lobe and an N lobe, ATP site in the middle, and the hinge on the left-hand side. So to actually generate starting points for inhibitor design, we screened a relatively small library, less than 4,000 compounds with a biochemical assay. And we selected a potent azoindole hit for optimization because of the hinge binding motif with a favorable IP position. And you can see that hit here on the left-hand side. We solved the X-ray structure of MPS1 bound to this compound, and you see it binding to the hinge here. It was very potent, 25 nanomolar, but the problem with the hit was that it was not selective, and it also hit other kinases, and it had very poor cell-based potency. So we set out on a structure-based drug design campaign, which is sort of represented by this arrow, to actually improve this hit, and that yielded this molecule over here. This is a very potent molecule, and it's now also selective and has cell-based potency. And structurally, what was interesting was actually that this Bock group here, that you see here, ordered or stabilized the activation loop in an inactive conformation that was sort of incompatible with ATP binding. So this all looked quite good. However, this molecule had a relatively high molecular weight. It had a high lipophilicity, and it had a labeled Bock group. and significant inhibition of P450 enzymes. So it was not fit for clinical development. However, as a chemical tool to actually unravel MPS I biology, it was okay. So what did we do then? Here you'd see that, that tool molecule again. We also had another hit for which we had the crystal structure that you see here, and that hit overlapped quite nicely with the binding of our tool molecule. So the idea was actually to scaffold hop to this hit and improve it further. And this allowed us to translate quite a lot of the SAR onto the new scaffold. Again, a structure-based drug design approach that yielded this molecule over here. And that molecule is now again quite potent, selective. It is of lower molecular weight, and it has a stable replacement for the Boc group over here, and it doesn't hit P450, I think. However, it was still metabolically labeled and not selective enough. So we improved it to ultimately to this molecule here. The methyl group here actually made it much more metabolically stable and more selective, as did this group over here that binds sort of over the hinge. Crystal structure, again, binding very similar to the other others. And this became our phase 1 clinical compound. It's an orally available, potent and selective MPS I inhibitor, and it showed sustained MPS I inhibition in a tumor model. So, however, we didn't stop here at the drug discovery, because one of the very big challenges in cancer treatment is acquired resistance through therapy. So already during the drug discovery, the project leader and biology lead, Spiros Linadopoulos, actually decided to try to identify potential MPS I resistance mutations. And resistance mutations in kinases quite often occur by mutations in the ATP binding site or in the kinase. So what he did, he grew HCT116 cancer cells, his colon cancer line, in the presence of two chemically diverse competitor MPS I inhibitors, an AstraZeneca molecule and a Nerviano molecule. And then he sequenced the MPS I gene for resistance mutations. Now that the AZ molecule gave 2-point mutations, which you can see over here, and the Nerviano molecule gave 3-point mutations, resistance mutations. And these 5 mutations conferred resistance against multiple MPS I inhibitors, including our own. So what he then did is he analyzed patient samples and identified all mutants in 14 breast cancer samples that had not seen treatment. In addition, he also identified these mutations at a very low level in normal healthy lymphoblast samples, and that was quite a shock to us at the time, because what that meant is actually that the mutations didn't actually occur during the treatment, but they were already present, and it was just actually applying a selective pressure on the drug of a drug that would actually lead to outgrowth of resistant clones. So this actually shows you that actually thinking about resistance in drug discovery, you'd better do early, and that is what we try to do. So, of course, as a structural biologist, we wanted to actually understand the molecular basis of the MPS I resistant mutations. And what we did in my team, and here you can see the mutations sort of indeed, they all map sort of in and around the binding site of ATP and also of the drugs. What we did is we attempted to generate these mutations recombinantly, crystallize them, and then determine crystal structures of wild type MPS I bound to the drug and the mutant bound to the drug. And for this serine to glycine mutations, what we could see is that the AstraZeneca drug actually bound in exactly the same manner. And from the structure, it wasn't immediately obvious what was happening. However, this serine, the serine in the wild type actually makes an helix capping hydrogen bond. And by removing that, what we think is happening is that it sort of destabilizes this unusual conformation of the activation loop, which also binds to the protein in this site. The second mutation was an isoleucine to methionine. We couldn't produce the protein here, but what we did is we modeled every possible rotamer of the methionine residue, and that is sort of represented by this gray sphere. And what you can see is that all the rotamers actually clash with the inhibitor. So it's probably a steric clash that conferred the resistance in this case. Now, the cysteine to tryptophan mutant, we were able to obtain sort of wild type and mutant crystal structures bound to the Nerviano compound. And what you can see is that in the mutant, the compound is shifted. And that is because of the clash of a CF2 group here with the tryptophan residue. So again, it's steric clashes that probably weaken the binding of this. This is further actually confirmed by a compound of our own, which doesn't have that CF2 group, that actually binds in exactly the same way in the mutant and in the wild type. So structural biology can provide insight in the molecular basis of resistance, and that is what we did here. So the second project I want to highlight, and you've already heard about it from Yannick earlier today, is our project to discover BCL6 inhibitors. Now, BCL6 is a transcriptional repressor, and it promotes antibody maturation by enabling clonal evolution of B cells. It suppresses regulators of cell cycle control, cell death, and DNA damage response. So it also enables germinal B cells to evade growth, checkpoint controls, and tolerate very high levels of DNA damage. And it binds to its co-repressors, N-core, B-core, and SMART via its BTB domain. You can see that here in this cartoon, and to the DNA via its zinc finger domain. So you can also see that here. So in a clinical context, aberrant expression of BCL6 makes it an oncogenic driver in more than 40% of diffuse large B-cell lymphomas. It also promotes resistance to tyrosine kinase inhibitors in leukemia, and is overexpressed in a number of solid tumor cell lines. So binding to the co-depressors, and this was published already before we started, is essential for the repressive and oncogenic BCL6 functions, and disruption of this BCL6 co-repressor interaction inhibits DLBCL cell growth. So this for us was, that this gave us confidence that actually we could initiate an inhibitor program to target this BCL6 co-repressor interaction and disrupt it. So here you'd see the domain organization of BCL6. The BTB domain is located on the N-terminal site, and then there is a huge sort of fairly unstructured region, and the DNA binding domain is located on the C-terminus. So here in the middle, you see the published crystal structure of the BTB domain dimer complex with its co-repressor SMART and B-core overlaid on top of each other. They bind in a very similar way, in a very elongated, shallow groove at the dimer interface. And because this is a dimer, it has two, one at the front and one at the back. So this, we figured, was a really challenging interaction to disrupt. So we would need a very comprehensive hit identification and validation approach. So this is what we did. We did virtual screening, we did high throughput screening, and we did fragment screening, and we followed all the hits up with a raft of biophysical techniques with the ultimate aim of characterizing the most promising hits using X-ray crystallography and actually elucidate the binding mode. So I'll now focus on the HTS because that is actually where we got the hits from that we ultimately progressed. We used 1536 FP assay with fluorescently labeled B-core peptide, and you can see that the data for that probe here. It binds with a 1.7 micromolar KD. And what you can also see here is that with unlabeled B-core and SMART, we could complete the probe off, and we used one of these peptides as a control in the screen. So we screened at 200,000 lead-like HTS library at the 50 micromolar final concentration, single point, and we obtained, after removal of interferers and non-available compounds, just over 1,000 primary hits. We then did a confirmation at 3 points on the same screening sample. We looked at the purity and kicked out the compounds that were not pure enough. We removed interferers again, and then did a full IC50 determination, which left us with 34 confirmed hits. At that point, we usually, and we did that in this case as well, repurchase the compounds. We could repurchase 27 of them, and then did a full IC50 determination again. That gave us 24 hits, and 23 had an IC50 that ranged between 70 and 700 micromolar, and one had a percentage inhibition of 34% at 1 millimolar. Now, this is very weak by HCS standards, so, but we are targeting a challenging interaction. So what we figured is that we needed a very robust hit validation protocol, and we used biophysics for that. So before we actually started the biophysics, we determined the solubility, because biophysical techniques, as you probably all know, are very sensitive to solubility. So we wanted to actually have an idea how our hits would perform. We found a range, some with good solubility, some with poor solubility, and about 6 compounds with a solubility less than 30 micromolar. So we figured we might not be able to actually test everything due to solubility constraints. So what we then did was we set up a thermal shift assay that's based on unfolding of BCL6 in the presence and absence of a ligand. And the idea, as many of you know, is that if actually the ligand binds, it stabilizes the protein, and then it unfolds at a higher temperature. And indeed, the positive control, the B core peptide, had a delta Tm of 6.5 degrees. We tested the 24 hits at 2 concentrations and found 16 hits with a reasonable shift, 1 degree to 3.5 degrees, and 8 hits with a small shift between 0 and 0.5 degrees. And what was reassuring, that there was reasonable agreement between the TSA results and the FP dose response experiments, with the biggest shifters actually also the compounds having the higher affinity in the FP assay. We then set up SPR, BCL6 BTB domain, immobilized using amine coupling. We were able, due to solubility, to characterize 19 hits. We determined the KD for 12 compounds, which ranged from 49 micromolar to 1 millimolar. And these are the 2 best compounds, and you can see they have reasonable sensorgrams and reasonable Langmuir binding curves. So, but TSA and SPR tell you that the compound binds, but it doesn't tell you where the compounds bind. So to get an initial insight in that, we decided to map the B core smart binding groove interaction hotspot with smaller peptides based on the co-repressors. And here at the top, you can see sort of the red bars. They sort of represent the tetrapeptides, overlapping tetrapeptides that we designed based on the smart peptides. And here at the bottom, you see blue overlapping tetrapeptides that we designed based on the B core peptides. And what we then did, we did a ligand observed NMR experiment, and we looked, and the binding was measured as a decrease in peak intensity. This was a CPMG experiment. So what we found was that the WVVP peptide was the smallest peptide with BCL6 binding affinity, and you can see that over here. And here you'd see the spectra. This is the WVVP on its own. And then if you add BCL6, you see this peaks disappear. That's a sign of binding. When you then also add the B core peptide, you see the peaks coming back up, which is a sign of that you're actually competing the tetrapeptide off with this longer peptide. So that was reassuring, but the WVVP peptide wasn't actually that easily synthesized. So we decided to generate a hybrid peptide, a WV IP peptide, and hybrid between SMART and B core, which showed similar behavior and was a lot easier to synthesize. Now, based on that WVIP peptide, we designed a 19F peptide NMR reporter probe, and we set up a 19F NMR displacement assay. So why would you want to do that? Because 19F is very sensitive to its chemical environment. It's not present in proteins, and it shows minimal spectral overlap. We tested all 17 hits that we used. We tested in the 17 hits that we tested all displaced the probe. And the percentage of displacement correlated again very nicely with the affinity in the FP assay. You can see that here on the right-hand side. So The hits bind in the WVP interaction hotspot within the co-repressor binding site. But of course, we didn't know anything about the exact binding mode. For that, you need structural biology. So BCl6 crystallizes quite easily. However, our initial crystals diffracted quite poorly, and it took us a little bit of time to obtain a robust, high-resolution BCl6 crystal system that was suitable to drive a drug discovery project. We needed to be able to do this routinely. I mean, and we did this by co-crystallizing it with the WVIP peptide that we used in the NMR experiments. Now, the WVIP peptide binds in 2 sites in the co-repressor binding groove. You can see that here in the middle structure. The top blue one actually binds in a crystal context, so we didn't think that was functionally relevant. But this pink one here binds in the middle of the groove. And we realized, or we checked, that it was back soakable. So by back soaking protocol, we managed to actually soak our HTS hits in, and all of the HTS hit bound exactly in the WVIP binding site, which was quite reassuring and backed up the NMR experiments. So we had quite a high confirmation rate or translation rate from the biophysical, the hits to the structure. And I think that's because we had a very rigorous biophysical confirmation and validation in front of it. So how, and all these hits, they mimic the co-repressor peptide interactions. And you can see 2 of them here. You've seen them before. These 2 were the SPR hits, the best SPR hits that we had. And you can see here, all the hits actually make this hydrogen bond between a compound and a backbone carbonyl atom of the protein. All the hits also stack between this tyrosine over here and an asparagine at the bottom, and the hits tend to have a group here, usually a ring system that lies against the backbone of the protein in this area, and you can see that here as well. This hit actually makes additional interactions. So, but the solubility was pretty poor. So the first thing that we did is try to improve the solubility. And we did that by reducing lipophilicity and removing aromatic rings. And that yielded compound 5 and compound 4. And you can see they have similar affinities as the original hits, but the solubility is a lot better. And their interactions are more or less, and the binding mode is the same as the original hits. So what did we do next? Well, you can see here that the left-hand side of the molecules is pretty similar. In compound 4, this group actually makes additional interactions, but in compound 5, the cyclopropyl group doesn't do very much. So what the idea was, it's not rocket science. We merge the compounds, and what we then hope is to get a compound with higher affinity that behaves better. And that yielded compound 6, and indeed, it had a higher activity, and the binding mode, all the interactions that were present in the original hits were replicated in this compound. So we selected this compound for hit optimization, although we lost a bit in solubility, I have to say. So this actually slide represents our structure-based hit optimization. This arrow represents a lot of medicinal chemistry. Now, what the idea was and what we did was actually we wanted to gain activity by extending the molecule in this direction, in this space here, in the BCl6 binding groove represented by this orange arrow, and to sort of exploit a hydrophobic patch sort of near this valine residue. And the other thing that we tried to do was actually optimize the stacking interaction of the left-hand side of the molecule. That ultimately yielded this compound 25B, and indeed you can see here is a group that actually exploit that space there, but what it also does is exploit a network of bound water molecules that actually helps to with the affinity. And then on the other hand, this moiety here, this group over here, doesn't really make polar interactions with the protein, but helps to optimize the stacking in this area. This gave us in the biochemical assay, now it's a TR-FRET, not an FP anymore, because this is more sensitive, the TR-FRET assay. This is a nanomolar compound, and also in the cellular target engagement nanobreath assay, it had submicromolar activity. So this is all very nice. And this is actually where it became quite interesting, because here you can see that again, this is compound 25B, a very similar compound with a slightly different sort of solvent exposed group over here. Was also potent in the biochemical assay, but actually was completely inactive in the nanobreath assay with a dose-dependent reduction in total luminescence. We thought that might suggest cellular toxicity or depletion of one of the assay components. And at this stage, it was at this time where Boehringer and Ingelheim disclosed their BCL6 compounds, and they had very similar groups present in the compounds, and they were BCL6 degraders. So obviously, what we then decided to do is test if our compound 24B also degraded BCL6. We dosed 2 diffuse large B-cell lymphoma cell lines with BCL6, and here you can see that in both cell lines we have dose-dependent degradation of BCL6. So obviously degradation is an attractive alternative to inhibition, so we decided to further pursue a degrader modality. So this involved further optimizing the compound and actually improving the clearance. And this yielded this compound, CCT373566. This is a very potent degrader with high binding affinity, and you can see the binding, the crystal structure here. It binds similar to the other compounds. It's improved clearance and exposure compared to our earliest degraders, and it shows sustained BCL6 degradation for more than 12 hours in a DLBCL xenograft model. However, so that looked all good. However, it only showed very modest in vivo efficacy in a DLBCL xenograft at 50 mg/kg/BO for 22 days. So this is actually where we stopped the program, because we couldn't improve on this, and we knew we were hitting the target reasonably hard. But then we stopped, but not quite, because everybody always asks me when I give this talk, how does these BCL6 degraders, actually, sorry for the typo here, work? Well, Boehringer and Ingelheim, together with the Fisher Lab in the United States, actually published a very elegant paper on and where they looked into that. What they observed that actually that in cells, when they dosed it with their BCL6 degraders, you got this BCL6 foci. And what they then did, they did some lovely cryo-EM, and they showed that actually their degrader polymerized the B2B domains into large filaments. And you can see that here in the electron micrograph, and here you can see the model. And the degrader actually glues the B2B domains together in these large filaments. So obviously we were quite interested if that would happen with our inhibitors or degraders. And obviously we had our BCL6 sort of dosed with our degrader in the electron microscope as well. And you can see here that indeed they form filaments. And if you look at the 2D class averages, you'd see a wide variety of assemblies, sort of 2 B2B domains glued together, more together, and even filaments that intertwine. What we also could do is actually dock the B2B dimer with our degrader in quite nicely. And here you can see the density for the degrader in between the 2 of the B2B domains. So both the BI and the ICR BCL6 degraders induce the formation of these BCL6 polymers. The polymers actually are recognized by CA1, the E3 ubiquitin ligase. That is what BI and the Fisher Lab found out as well, and that causes proteasomal degradation of BCL6. So here again, sort of structural biology can help you to elucidate the mode of action of your compounds. So this leads me to actually talk a little bit about the increased use of cryo-EM in drug discovery at ICR. So for us and for my group, the challenge is that our targets that we typically work on in cancer tend to be mostly soluble, no membrane proteins, and small and mid-sized. so not that suitable for cryo-EM. So where do we deploy cryo-EM and small molecule cancer drug discovery then? Well, we use it, and we have used it successfully now when the target cannot be easily crystallized, or the crystals actually don't diffract very, very well. And in this case, this cryo-EM can really help you out. We also have examples where the inhibitor-bound conformation of the target cannot be easily captured in a crystal system, and this is also a way where cryo-EM can help you then. And we also use it, of course, in characterizing compound binding to a multi-domain protein or multi-protein complexes. And this is how I started establishing it at ICR. I established the cryo-EM in the protein degradation space. And here you can see an example of one of our earlier structures that shows the DDB1 cereblon complex with an IMiD bound and a zinc finger protein that we were interested at the time. So what helps me and my team is very much that we're firmly embedded within the division of structural biology, because we can make use of their infrastructure, the microscopes, but also the methods and the state-of-the-art thinking in the basic science structural biology world. So now one of the final slides, how do we use AI and Alphafold at ICR? We played a little bit with Boltz-2, which is an open source protein ligand co-folding software, and that is claimed to predict ligand deficiencies with an accuracy that approaches free energy perturbation methods. Now, I think our view, and I guess, and well, we'll hear it in the panel discussion, and I guess the view in the structural biology world is that, yes, it can get poses right, but ranking affinities, it's not yet good enough for that. And that is because there's not enough protein ligand structures with affinity data associated with this to train the co-folding models, and we've heard that earlier today already. So large efforts have been initiated now to generate this data, for example, the Diamond-led Open Bind initiative. So Alphafold is a different matter, I think. Alphafold we almost use on a daily basis, and is very much integrated, mainly in construct design and structure determination for SPDD. So what is actually it's very useful in is to predict parts of the protein that are closely linked together, domains that are associated that you wouldn't necessarily expect, or parts of the structure, and that helps us to actually generate stable construct quite quickly and quite easily. Obviously, what we also do is phase experimental X-ray structures with the Alphafold models, and we fit protein models of Alphafold protein models into cryo-EM electron density, like probably many other people. So in summary, I hope to have shown you that the drug discovery project at ICR are underpinned by robust target validation in our new Center for Target Validation, and this helps us to select the most promising targets to work on. The Center for Protein Degradation extends our range of modalities that we can actually use to target our targets of interest, and builds on our already our vast expertise in small molecule drug discovery. I hope that the MPS1 and BCL6 projects show how integrated hit ID and follow-up campaigns can successfully drive a structure-based drug design approach. Cryo-EM, I mean, obviously it's going to stay, and it's now fully embedded in our workflows, and we use it now sort of almost in parallel with setting up X-ray. And Alphafold and AI have been integrated in CCDD workflows, as I said, mainly in construct design at the moment, but we will see an increased impact on small molecule drug discovery for sure. Final note, acquired resistance remains a big challenge in cancer drug discovery, regardless of the therapeutic modality of targeting your target. So this brings me to the acknowledgements. Of course, drug discovery is team science, and you cannot do that. You cannot discover a drug on your own. So we had vast chemistry teams on the two projects. The BCL6 project was led by Sven Holder, and the MPS1 project by Julian Black. The biology of the MPS1 was led by Spiros Linadopoulos, and the biology of BCL6 was led by Olivia Rosanes. And my team actually supported both projects with assays, screening, biophysics, structural biology, protein production, you name it. So now, in a final word, I'd like to acknowledge the synchrotrons, particularly Diamond and ESRF, where we collect most of our data. And I mean, these have been extremely helpful, and I don't know where we would be without them. So thank you for your attention, and I'm open to actually answer any questions.
So thank you very much, Rob, for this insightful talk. Thank you. Are there any questions from the audience?
Yeah, Jose?
Wait.
Oh, you have to wait for the microphone.
Hi, Jose here from Diamond. Rob, you mentioned you had started with many fronts, ended up with the high throughput screening one. Comparing to fragments, is it just because you got the high throughput screening, you got on from there because you had the lead-like compound?
Well, actually, I mean, you know, in the drug discovery project, some things read out at different times. Actually, we were working for a long time on a virtual screening hit before we had but that actually plateaued, and then we moved on to our HTS. The fragments, they just came a bit too late in the game, and then because they're so much weaker, although these are weak HTS hits, the chemistry had moved on at that time, so it was just a little bit too late in the process.
Okay.
But we had quite a lot of fragment hits, and not only in that particular binding site, also somewhere else.
Thank you.
We had one question on Slack, which might be interesting because there are many students here. And could you maybe briefly explain the concept of back soaking?
Back soaking. Okay. So what we'd like to do, and what is the easiest to actually in protein ligand drug discovery, is if you have your protein crystals and you can just soak them in a solution with your inhibitor. The inhibitor finds its way through the solvent channels to the binding site, and it binds, and then you can actually solve the structure. But sometimes it's very difficult to get crystal structures with an apo active site, and you have to have something in it, whether that's sort of ADP and or a peptide or an inhibitor. And then what you want to do is actually get that out first, and then soak your compound in. And this is a process we call back soaking. You soak your crystal in a solution that doesn't contain anything, and then you'd hope that your ADP, or in this case, our tetrapeptide leaches out. We sometimes do it directly in combination with the inhibitor of interest, and we hope then that, because the inhibitor is hopefully somewhat more potent, that the inhibitor replaces the compound that was originally in there. So it's a trick to actually get to protein ligand structures. Doesn't always work, especially not if actually the ADP or the other compound that you originally use actually binds very tightly in the binding site.
You have one question here?
Thanks, Rob. That's a very interesting talk. So I'm pretty interested in the MPS1 inhibitor developing story. So as you say, you have lots of hits binding at the catalytic pocket, which is at the hinge region. Mm-hmm. Then you merge these compounds into a lead compound. That's quite interesting. My question is, how did you optimize those leads to give them a better selectivity? Because if the compounds bind to the catalytic pocket, it's a very big question. Is they sometimes can be quite promiscuous and bind to lots of different kinases. So in your case, how did you optimize the selectivity?
So the activity was optimized by filling out the pocket, essentially, and by making them bigger. You can do that. We extended it. The selectivity actually was quite important. Some kinases have a small residue in the hinge. I think it's the gatekeeper +2 residue. Some have a bigger. And actually, the ones that have a small hinge residue there is a small pocket usually that you can exploit, and that is exactly what we did in MPS1 kinase. And also, we looked at other kinases, we overlaid them with other kinase structures, and then I think for a clinical candidate, the methyl group that we added onto the core scaffold also showed from docking studies and so on, that showed that we could gain selectivity there. Does that answer your question? Okay.
Any more questions? Can't see any. Please use Slack if you have any more questions. I think, Rob, you're around for a while.
Yeah.